Thank you very much, Damian, for the introduction, and thanks a lot to the organizers for giving me the chance to explain my work and my research here today. So as Damian was saying, I will talk about integer topological defects and how they direct multicellular organization in tissues. I actually work with cell monolayers, so like the easy version of a tissue. But I will start with the word nematic. I'm sure you all know a little bit about this concept, but maybe you're not so sure about its origin. And actually it was invented to describe these phases, which are nematic phases in thermotropic liquid crystals. And I started working with this. So my beginning in research was actually working with like passive liquid crystals and nematic phases. And these are basically fluids, so they behave like a liquid. If you see them, like you see that they flow apparently like a normal liquid, but they present like molecular order because of the nature, the chemistry of these molecules and the physical shape of the molecules. These molecules interact with each other and give rise to long-range orientational order. And this is very important for the physical properties that arise from this material. I mean, you all see liquid crystals every day through your screens or your devices, and probably they don't have these like blades or these shapes that you see here. This is actually, when you perturb a liquid crystal, this is what you see through the microscope. So this is an out of equilibrium version of a passive liquid crystal. And you see these black and white parts, and this is because the light interferes differently with these different regions, depending on how the molecules are oriented. And another important feature of liquid crystals are topological defects, which are these black crosses you see here. And these are regions that are required and necessary to link regions without different orientations. And this is actually very important in biology. And what we are trying to do now in the labs are analogs of these liquid crystals that are activated at a very local scale. So prepare liquid crystals from units that are active. And these are called active liquid crystals or active nematics. I think the most beautiful one, in my opinion, is this one. And I had the privilege to work with this material. This is made out of, it's composed of microtubules and molecular motors. These are filamentous proteins and that we grow artificially in the lab. And we cross-link these proteins with molecular motors that shear these microtubules locally. So basically by consuming ATP, the system is continuously fed with activity at the local scale. And these are the patterns we see when these systems are active. This is a quasi two-dimensional system, and it's active. As you see, it features these like classical non-equilibrium patterns like we see in, for example, fish flocks fish schools or bird flocks. This is called actually active turbulence because it's very reminiscent of like classical turbulence, but it's really happening at really low Reynolds numbers. So it's these systems are really viscous and the inertial forces are really slow, really small actually. So the velocities are very small. So this is called active nematic, and you see that it has like black things that move around. These are topological defects as well. Interestingly, these topological defects are now active. So they exert forces and they move because they exert forces to their surroundings. And actually, yeah, these are the two defects we are seeing here, +1/2 and -1/2 defects. This number is the topological charge. And we put this number just by looking at how the director field or the nematic field is organized around these singularities. Okay, so if you moved like a test rod around the core of the defect, you would see that the rod turns 180 degrees in both cases, but in one case it turns in the same sense of the rotation, in the other one in the opposite sense. If you need like clarification on this, please interrupt me at any moment. But basically, this is the numbers we give to the defects to describe them. Actually, you see that the -1/2 is symmetric. So this wouldn't move, but it moves because it's advected and moved around because of the presence of the +1/2 defects. And these defects are created all the time, and this is what gives this kind of turbulent state. Okay. So actually, the presence of these defects we are seeing now that are really relevant for biological processes and tissue homeostasis. So tissues need to make certain processes to deform, to maintain itself like in good shape. And one of these processes is cell extrusion. So cells need to be extruded from monolayers. And actually, recently, Benoit Ladou's group saw that the position of these topological defects in cell monolayers correlates with extrusion of cells out of the monolayer. And actually, this is because these defects have always the same orientation around them. And this provides with a very precise mechanical pattern that allows for the cell surrounding one particular cell to extrude the cell. So basically, these defects, what are doing is compressing a particular region and extruding cells outside the monolayer. So as I said, these nematic phases and these topological defects are really relevant for biology. These are epithelial cells, but they have been observed as well in monolayers of neurons, neural cells. And actually, the position of defects here correlates with like changes in density in the monolayer. So actually, biology is using topology to organize cells in a tissue. And this is something I think really striking. And actually, the last, most recent paper describing defects in biology is related with Hydra. These are really weird bugs that we have in freshwater, actually. These are really easy to get. So probably we drink these things when we go to the mountain and take water from the rivers, but it's just more protein, so it's nothing bad to eat. It just moves around, and it's really spectacular because this is probably the multicellular organism, the simplest one, the simplest multicellular organism that we have on Earth, actually, one of the simplest. And it's composed of two layers of actin that present like striking nematic patterns and topological defects. And actually, I was like really inspired by the previous talk by Anton, describing like these shapes in active shells. And actually, this is really reminiscent of how morphogenesis works in biology. So you start with a ball of cells that deforms in different shapes because of different chemical and mechanical patterns that this system is subjected to. And this Hydra actually deforms, giving rise to defects, and deforms from a sphere. So a sphere, as you probably know, it's a spherical shell actually. It needs a topological charge of +2. This is like set by mathematics, but biology obeys these laws. So Hydra actually is topologically stable. It generates defects to create the mouth, the feet, and the tentacles, and this is topological stable. And I think this is pretty striking. But actually generating these defects and controlling them is not so easy. So this was a little bit our obsession. So as you see here, there's another defect that arises, which is the integer defect. So I've talked about half integer defects. There are integer defects as well, but they're really energetic. So it's really difficult to get them spontaneously in unconstrained active systems. So to get them, you really need to constrain somehow your system. But these defects are really interesting because they are non-motile, but they are active. So all the forces are basically being directed to processes that are not motion, but this doesn't mean that it's not active. So we have active defects, but they are non-motile. So the forces go somewhere else. For example, here the defects can open a mouth or grow a feet or be used to adhere to the surface. So this is how Hydra uses the +1 defects in this case. So we were a little bit, this was our objective. So we wanted to create integer topological defects within cell monolayers and see if they could result in like relevant biological processes. So to do this, we do this via confinement, and this is probably the best tool we have to control active systems, and especially if they are topological materials. So if they have to obey certain topological rules and to geometrical constraints, here's where confinement is really relevant. So actually, passive liquid crystals can be controlled by confinement. So confinement would impose certain topological constraints, and we will generate topological defects to satisfy these constraints. And we can do this with active systems. We just need to know exactly the relevant length scales of the active systems and confine them between below confinement sizes of the order of this length scale. Okay. So this is what we did with cellular nematics. I'm going to introduce you to my active system. It is composed of muscle cells. These are really strong cells. They are really contractile. and they perform mesenchymal migration, which is basically they slide one past each other. So they have really low cell-cell adhesions compared to epithelial monolayers, for example. And as you see, they have like really, really anisometric shapes. And that's why we get nematic phases out of them. So what you're seeing here is probably the easiest experiment you can do with cells. You just plate them on a glass surface coated with their favorite protein. And they basically start spreading. And you see that they elongate, and slowly they will interact with each other and generate these kind of nematic phases. So generating long-range orientational order and topological defects. This is the unconstrained case. What we did with this is basically characterize orientation and velocity after imaging. So we do image processing and perform velocimetry and orientation analysis to get the position of the topological defects, the velocity patterns, etc. And we can get like a lot of different quantities to characterize these as an active material. So the first thing we did is get this orientation, get the position of the defects, get the relevant length scale. As you see, this looks exactly like a liquid crystal. It would look at the molecular scale. So we have minus 1/2 defects plus 1/2 defects, this long range orientation, etc. So the first thing we did is characterize the relevant length scales of the system. In time, you see that it increases in time because these nematic phases do not appear at the beginning. They appear when the cells interact with each other. And this length scale of 200 micrometers more or less is the relevant length scale, nematic length scale for this material. So what we did is we start confining these systems in circular geometries. And the way to do this is using micropatterning. So basically we cover a surface with polyethylene glycol that cells hate. So cells wouldn't adhere to the blue part of the surface. And then we burn this speck and we add fibronectin, which is cells are super happy on fibronectin, so they will attach on fibronectin and just start dividing and proliferating on top of these circular islands. So the two topologies you can expect from circular confinement is these two kinds of defects, and in the middle you have like infinite spirals in between these two configurations. And basically what you're changing here is the alignment of the cells with the boundary. We impose like really strong boundaries, and this propagates inside and generates integer defects. Okay, so the first time we did this, we were a little bit, we were expecting like a very striking result, and basically what we see is that we get like rotation, cells aligning parallel to the boundaries, and at some point cells stop rotating and they align perpendicular to the boundaries. This happens within like 40 hours, more or less. Cells are quite slow, so you have to image them for long. But basically what we are seeing here is a transition from ring, spirals, and aster defects. And these are like very common defects in liquid crystals. So we are studying these cell monolayer islands from this perspective. So we see collective migration, two migration modes actually. We see rotation and a static mode, and then we see two orientational configurations which are corresponding to a spiral and an aster. So what we can do is we can study these like more in detail. We can extract velocity and orientational fields. I think my presentation just froze. No, now it's working. And we can get this transition, we can get the parameters, the evolution of relevant parameters like vorticity or the psi angle, which is the angle of the cells with the radial direction in time. And you see that there's a clear transition from something that rotates and it's anchored parallel to the surface to something that is like rather static, so it's not rotating anymore. And the cells, you see that they align perpendicular to the boundary. So this is a transition from a spiral to an aster, which is actually predicted from theory. So we're happy to see this. So we can stabilize spirals, for example, by blocking proliferation. So we've seen that this transition happens because of density. So if we block proliferation, basically we stabilize cellular spirals. And if we let, yeah, as I said, we can characterize orientation and speed. And if we let these systems to evolve for longer, this density increases and we get like cellular asters that are really, really static. But as I said, they are active. So these cells are performing, they are exerting forces on the substrate and within the monolayer. And we were wondering what these forces are doing. And what we did is we well, we characterize these two orientations, the spiral, sorry, these two configurations, the spiral and the aster. And we teamed up with Carlos Blanc from Karsten Cruz's lab in Geneva to feed some of these parameters to their theory. Actually, yeah, here there are more details, but for the sake of time, I will just skip this. If you have any questions about the parameters we've analyzed, I mean, I'll be happy to discuss later. What we've done, we fit some of these radial profiles for different sizes of spirals to Carlos fit his theory. Actually, this is a hydrodynamic theory for cytoskeletal active gels, actually. But Carlos has introduced like some terms to introduce traction forces and isotropic stresses within the tissue, etc. And this fits pretty well. There are some things that we are not so happy about, but this was enough so that Carlos could impose the material properties that he would get from spirals to asters and predict compressive forces. So this is explained in these two papers that were published at the beginning of this year, where the theory is like well explained, and we can get like a wide variety of like active phases depending on if traction forces dominate or internal stresses dominate, etc. Out of these, I mean, I think the most relevant thing that I want to say today is these compressive forces. So if you have an aster of this material, you would compress the center of these islands. So we get really high compressive stresses we predict compressive stresses at the center of these asters. And actually nuclear density should increase as well in the center. And actually when we look at these islands for longer, we see something that is really going along with these predictions. We see increasing density in the center, and we see somehow like protrusions at the center of these islands. We wanted to study this a little bit more in detail. Actually, we wanted to clearly see with our eyes if these forces were so strong that as they were supposed to be from the theory. So that's why I thought about this system. So these cells, they can heal like big holes. So basically we can generate the same asters I was talking about with a hole in the middle. And before they close the hole, what I can do, I can basically put a pillar in the center made of like elastic material that is really, really soft. And you get something like this. So you get a ring of cells around an elastic hydrogel pillar. And basically cells will close this hole and compress the pillar. And from this compression, actually, we can directly get the forces that the cells are exerting in the center of the island. So you see that the cells are really, really strong. This pillar is like around 4 kilopascal. It's like a little bit softer than Jello, I would say. So to characterize this a little bit better, we made the pillars fluorescent so that we could get like the compressive forces in 3D and get a better characterization of this compression. And this is like a steady image of a pillar compressed after a while by these cells. So you see that the pillars are really squeezed by the cells. And we can do this as well in time. So you see compression in time. We can get actually the forces that the cells are exerting around the pillar in time. So the other thing that theory predicts is not only like a peak of compression in the center of these islands, but a gradient of tension. And this was not so clear for us. So what we thought about, instead of like putting a pillar in the center and that would be squeezed, we just put a pillar off center, like so that in principle, if there's a gradient of stress or a gradient of pressure, this pillar should be tilted. Okay, and actually this is what we see. If we go cells around this pillar, we see that the pillars are totally bent towards the center of the pillar where it pops out, basically. So here's where the pressure is maximum, but because of this gradient of pressure, this pillar is bent towards the center. So basically, with this experiment, we report the compressive forces that these cell monolayers, now they are not that much a monolayer, they are more like a mound. I will explain this in a second. But we can report the forces, the compressive forces that these configurations do, and the pressure gradients that are supposed to arise in these preparations. So an interesting question that I think it's more relevant maybe for biology is how these forces are dissipated in a tissue. So how is biology using this kind of configurations to modify tissues or to make tissues evolve in a certain way? So one thing I've not explained to you is that these cells differentiate. This means that they engage into a pathway to express a certain kind of protein and to become a cell that will be able to make a certain function. In this case, I'm using muscle cells, and these cells are really going to be a muscle in your body. So you have these cells and they make muscle in your bodies. And if you have a wound in your muscle, they will just go out of your muscles and heal the wound and then make a proper muscle. And the way to do this, they need to align, as you see here. They need to fuse, and they need to express myosin, a protein that is like a classical protein for contractile networks. And this is how you make muscle fibers. And actually, these cells do this. So what you see here are myotubes. So if we culture these cells like for a long time, you see these yellow things. What we stained here is myosin. So they are making muscle fibers. And this differentiation needs to be triggered somehow in the tissues. And we saw that actually these aster topological defects, what they are doing is localizing differentiation at the very center. So where we have the maximum compression is where these cells are activated for differentiation. The left image is probably the most beautiful one, but here you have like an average of like several of these islands, and you see that differentiation is happening preferentially at the center where we have the maximum compression. We can change these cells a little bit, so we can transform them so that they don't differentiate, they just proliferate. This is what a cancer would do, for example. Cancer cells, they don't differentiate, they just proliferate, they make tumors and they make tissues function in the wrong way. And actually, from these topological defects, we see that these protrusions arise. This is actually really striking. So in biology, it's not known of any experiment that you get 3D structures from a 2D layer. And actually, only by confining the system and generating these integer topological defects with one single cell line, we can generate shapes that are not spheres. So this is pretty cool. So we have these two cell lines. The left one will differentiate.
Oh, sorry, Pau. You are kind of running out of time. I mean, you can finish.
I will speed up a little bit. I think I'm finishing. So we get these two outputs, basically, differentiation or growth. And these are like weird shapes that I'm getting lately. If I prepare my patterns correctly, I can get these super long protrusions. They twist, they spin. They're really cool. And this opens, I think, a new venue in the physics of 3D tissues. And I hope I can explain you more the next time. The nice thing we are seeing is that this long-range orientation is preserved in 3D. So we are getting 3D cellular nematics. We get like a disordered region on the top, which we correlate with topological defects. The topology of this is a little bit more complex than a 2D disk, but we are working on this. And I think I'm finishing here. So this is like the summary. Sorry. Yes. So basically we prepare cellular nematics and integer topological defects by confinement. And this results in like localized mechanical stress that generates, certain specific supracellular organization. And yeah, I'd like to thank you for your attention. And of course, my previous lab members in the WHO lab and Carlos Blanc in the cruises lab. And this is a movie of us in a ski resort in France, actually, not Switzerland. And I convinced my lab mates to make a spiral and an aster on the snow. So this is like a human spiral to aster transition. Yeah, so you see a spiral here. The drone is rotating, not us, but I think it's good enough. And this is an aster. And yeah, I'd like to thank you very much for your attention. And that's all from me. Thank you very much.
Okay, thanks, Pau. Beautiful experiments. Time for questions. Just.
Yes, thank you very much, Pau. This is a very interesting talk. This fiber running out of your rotating system, it reminds me of the rod climbing effect in viscoelastic fluids which are driven. Have you tried to make an analogy with that in terms of an active viscoelastic fluid exhibiting this kind of behavior?
Not really. So yeah, thank you for the analogy. I mean, I've not thought about this, but it's a good analogy. I mean, this is quite recent, and we are trying to study how these things form. The main driving force I would say is the compression at the base. And remember, I mean, I've not said this, but these systems are proliferating all the time. So all this mass that is being generated is located in preferentially at the center because of this mechanical pattern. So this is for protrusion. Then the stability of the protrusion and the shape probably it arises from maybe a very fundamental. It has a very fundamental explanation, but we've not thought about this like. But yeah, thank you. I mean, it's a good analogy, indeed. I don't know if you were talking about like more the shape or the formation of the protrusion.
The shape and possibly, you know, as a simple toy model, you could think of that in terms of an active stresses acting on such a fluid, and then see if you can capture the generic features. But I think that's a topic for a longer discussion at some other point.
Yeah, but thank you. I mean, you're welcome to, I mean, if you want to try to get these shapes, I mean, it would be super cool. I mean, I've not seen this in simulations or theories, so this is a challenge for you all.
Okay, thanks. We have another question. Jan?
Yeah, thanks. I was wondering, is this differentiation that you showed in the center, is it caused by the pressure or the density of the cells? So, for example, if I took a single cell and compress it, could I induce the differentiation?
Yeah, that's a good point. Actually, we don't know. So we know that there's pressure, and we know that there's high density. Both could be driving this cell fusion and this differentiation. There are some experiments, though, with like monolayers of cells. They stretch them. That's the same cells. They stretch them, they compress them, and they see the same. They see that when they compress them, they have differentiation. So there, the density. Of course, it's difficult to conserve density in space. So it's difficult to distinguish which is the driving force. But I think you need both. So I'm sure the high density is as well a cause for the high pressure. So I think they are connected.
Thank you.